We have already discussed about isotopes. We have already talked about carbon dating and strontium. So we have already like gone through the two very important aspects of bioarchaeology. And here in the third presentation, we'll be touching on another aspect, which is archaeogenetics. And why am I saying that we are going to touch this aspect, or why is it an introduction to archaeogenetics and not as lengthy as the earlier lectures were, is because archaeogenetics in itself is a field that is developing. It's a field which has a number of debates going through them, and it is a field which requires a very sound theoretical understanding. With that being said, it's not like the lecture will not have any explanation part. It's not like the lecture will not talk about the basics. We'll be rather focusing on the basics and not going on to the more complicated analytical parts, at least in this lecture. And the basic content of this presentation is going to be focusing on genetics and how they are used in archaeology, then discussing about three very important discoveries that took place with the aid of DNA studies in archaeology. And then we will end our presentation with the flip side of DNA studies, and there could be no better case study than our own country, because DNA studies have revealed here that why archaeogenetics, although a powerful tool, is also something that has to be used with utmost caution. So, this is what the presentation is going to be about. First of all, let us understand genetics. So, we all know that a human cell, the core of a human cell is comprised of the nucleus. And in the nucleus, there are 23 pair of chromosomes, which makes it 46 in total. And either of the chromosomes are borrowed from the parents. So there'll be a maternal line of chromosomes and then be a paternal line of chromosomes. Together, they form the 23 pairs or 46 chromosomes. And a genome, if I go by the definition given by the National Health Organization in 2025, is the entire set of DNA instructions found within a cell. In humans, the genome consists of these 23 pair of chromosomes, while these are located in the cell's nucleus. They are also present in small quantity in the cell's mitochondria. A genome contains all the genetic information of an individual, and that's basically what the definition talks about. And genetics relies on the study of these genomes, of these chromosomes. Now, the chromosomes are further divided into nucleotides, and these nucleotides are organized in twin chains, and they are 3 billion in number. 3 billion on one side. So there's a pair. So these are 6 billion in total. And let me just show this diagrammatically. So you have the nucleus. The nucleus has the chromosomes in them, and the chromosomes, each chromosome has a gene pair associated with it. So this becomes your genetic material that we focus on in the genetic studies. Now, just a second. So these nucleoids are represented by sequences of letters or alphabets, and these letter alphabets, these form chain of letters. So if you think about it, there are 3 billion a pair of 3 billion nucleoids. These will form a chain of 1,000 letters, and every individual's genetic makeup is actually made of these chains of letter sequences. So this is a more kiddish diagram, but if we go into more significant diagrams, they actually explain this entire genetic flowchart. So you have the human cell. In the human cell, there's the nucleus, and then there are 23 pair of chromosomes, the paternal and the maternal, coming from both the sides. Then there are genomes that come from the paternal and the maternal side, and each of these genomes have 3 billion pair of nucleotides. They are represented by the letters TGCA. So there's like a sequence of letters TGCA, ACGT, GCATA. I mean, this is what the sequence look like in the 3 billion pair of nucleotides. And that's the very basic, fundamental, unitary component of genetics that geneticists or archaeogenetists study in archaeology from the present population as well as DNA samples extracted from the past population to understand about an individual, about where interbreeding could have occurred, about where the individual would have been associated with another species in case of prehistory. So that's where we stop with the components and focus on mutations. I hope I'm clear here. You can take a screenshot of this diagram. This is like the one diagram that I always refer back to when I forget the basics or the fundamental aspects of genetics. So, what are mutations? Now, as said, in these long lines of nucleoids, they are almost similar in all of us individuals, except for a very minute difference arising in the later parts of the nucleus of the nucleoid material. So this difference occurs because of copying of genomes from one generation to another. So when an offspring takes in their DNA from the father's side as well as the mother's side, almost the entire DNA material gets replicated in the offspring. But then there are minute errors that occur, and these minute errors in copying are called mutations. So if yeah, okay, so as you can see, this is like the father side of DNA nucleotide, as you can see, and the mother side of nucleotide present. So it will be almost same for all of us. So T G C A G C A G G C, but in the last, as you can see, there's a difference. So there's a T mutation coming, T nucleotide coming from the paternal side, but from the maternal side it is A, and this difference is called mutation. Um, in present day human beings, so like in our case, 99% of letters are same, as you can see. It's only just the 0.1% that is different, and that is also something which gives an individual their own unique genetic makeup. So I don't know if you would have seen this in movies or telesoap operas where they try to understand like who is the father or the mother of the child. They go for DNA test. This is what they are actually trying to do. This is what they are trying to see. They'll take the DNA from both the parents and from the offspring, and then they will look at the letters and see where the difference in the letter arises or where the similarity arises, how similar they are, how different they are, to understand if they are the offspring of the given couple. Another important point that has to be considered here is that the more differences that exist in two set of genomes, the longer it has been since they shared a common ancestor. What it exactly means is, think about this. So you have DNA coming from both the parents' sides. So you have the mother's side, the father's side. Now, if you bifurcate this figure into two parts and look at the earlier part, so you can see that there is genetic difference. Is the cursor visible? Yeah, okay. So you can see that there is a difference in nucleotides, but it is quite less in number. So more or less, the DNA material is very same between both the parents, which means that their common ancestor would have existed very close. That means the split that would have occurred, it would have occurred in a very recent time manner. In this case, it's 50,000 years ago. So, because most of us, now, why do most of us have this 0.01% or 0.1% of difference in our DNA? It's because all our common ancestors actually are very close. So the Homo sapiens or the Homo sapien population from which the entire 6 billion people on the globe derive today have their ancestor very close in time, and that is the reason why our DNA material is identical um, in all the individuals, and there's just minute difference, which is the unique difference. But let us look at the latter part of the diagram. So here you can see that there are a number of differences occurring. So rarely any of the alphabets, as you can see, they are congruent with one another. And this occurs when the ancestor that would have existed would have branched off years and years ago. So there is a long time since the common ancestor existed. Um, if you think about this in such a manner, so let's say you have a Neanderthal DNA extracted from some Neanderthal skeleton, and then you have the DNA of a Homo sapien existing almost at the same time, so maybe 50,000 years ago, and you would look at their DNA, so you would see that there are differences. But if again I take the human DNA, the Homo sapien DNA, and I compare it with, let's say, an Australopithecine DNA, there'll be more differences. Why? Because Australopithecines branched way earlier than the genus Homo did, and then when we go ahead in the history of the genus Homo, that's when um, your Homo sapiens, Homo neanderthals, Denisovans, erectus, habilis, they branched off. So the differences between a Neanderthal and a Homo sapien will be quite less when compared to the DNA of an Australopithecine and a modern human. So, okay, this is a recapitulation. I am not going through this part in detail again because we have done this in the first two lectures. DNA is extracted from the organic part of the bone. So, fossils are stones. Fossils are not organic in nature, so you cannot extract DNA from them. But if the bone is still in its decomposing stage, um, you can have the organic material present and you can extract DNA from it. Again, it's organic in nature. DNA is prone to contamination. It is also true that if it's not just in the excavation stage, but also in the laboratory that DNA can be contaminated. The microbial growth in DNA in certain cases is to the extent that only 0.2% or maybe 0.5% of DNA that has not been contaminated, the uncontaminated part can be extracted. So it's again a very tricky affair to extract a sample DNA, and then it has to be pre-treated for any sort of lab analysis, mechanical, chemical, and the researchers have to be very careful to not create any sort of biological contamination even in the laboratories. So this I think is very clear. um, Now we'll just quickly go through the three important discoveries or the three important studies that were conducted through DNA. The first one was Neanderthal admixture. So Neanderthals, as we know, are an extinct species of the genus Homo that existed in Europe and Southwestern and Central Asia almost 400 to 40,000 years ago. They are called Neanderthals because they were discovered in the Neander Valley of Germany in 1829. And as you would have read about it or come across it, they were quite intelligent species of the genus. So they made tools, they controlled fire, they had shelters, hunted animals, wore clothes. They also buried their dead and made objects which could have any artistic or ritual purpose. So, a question that does arise is that why did the Neanderthals disappear on the face of Earth if they were adept in almost all cognitive and reflexive functions? Then what caused their extinction? So, a number of archaeologists and anthropologists have suggested that when the Homo sapien population came out of Africa, if we believe the out of Africa model, if they came out of Africa, so they colonized the areas which were earlier occupied by Neanderthals, and they either killed them they um, they just made sure that they disappeared from the face of Earth. And this is what has been suggested. So this is quite a violent clash that took place between the two species, and our species emerged victorious. If you would have even read Yuval Noah Harari's *Sapiens*, you know what I'm talking about because he goes through that in detail. So it was because of social organization and the ability of human beings or our species. If I'm talking about, when I say human beings, I'm talking about our species to organize a larger section of people, to conduct group activities, and all of that. So, yes. There can be explanations, a number of explanations, but DNA studies have showed that Neanderthals never really disappeared from the face of Earth. They in fact exist today in form of our DNA sequence or in our own DNA material. So if you are someone who has been living outside Africa or is a non-African, you would have at least a certain percentage of Neanderthal DNA present in themselves. So all of us have Neanderthal DNA. Definitely, the percentage varies. In certain modern humans, the percentage can even can go up to 25%. And now, DNA, because it is extracted from both the mother's side and from the father's side, and it is also present in the mitochondria. So, one set of analysis in DNA studies is to look at this mitochondrial DNA, which represents the mother's side of DNA's communication. So, when I say that I'm looking at mitochondrial DNA, I'm talking about the female ancestry that I'm tracking. So, it confirmed that the common ancestor of the Homo sapiens and the Neanderthals existed 470,000 years ago, which does make a lot of sense, but it does not tell us if interbreeding did occur. Why? Because 470,000 years ago, Neanderthals were already in existence 400,000 years ago. And we know that we have descended from a common population. So if another 70,000 years ago to already 400,000 years ago, there was a common ancestor, that does not really surprise us. So what I'm trying to say here is that when was the time that the Neanderthals actually interbred with the species, with our own species, is something that the geneticists were trying to decipher or were trying to look at. Because when the two species separated, so you have Homo neanderthals and you have Homo sapiens, when did they actually interbred again? And they did this through a full DNA analysis. A full DNA analysis is an analytical technique where the mitochondrial DNA and the other sort of DNA is Y DNA. So that's gathered from the Y chromosome. You know about the sex chromosomes, XX and XY. So the Y chromosome actually tells us about the paternal line of ancestry. So mitochondrial DNA tells us about the maternal line, and the Y-DNA tells us about the paternal line of ancestry. And for a really long time, like towards the end of the 20th century, when DNA studies were coming to the fore, researchers actually used these fragmented pieces of DNA analysis, mitochondrial DNA and Y-DNA. But in the most recent studies of DNA, or the most recent development has been to look at full DNA, to get a larger picture, to get a more larger as well as a more sound picture of DNA studies. So they did a four population test. What is a four population test? I know this is getting a bit more complicated, but just hang on. It's nothing. It's just taking in four populations. So samples, DNA samples from four different kind of species or genus can be taken. So in this case, they took the chimpanzee genome, the Neanderthal genome, present day European and present day African genome. And what they did was they laid out the genetic sequence. So the letters, the 3 billion letters, they laid out for all of these different species, or I should say genus. And then they checked that at a given time, at a given period of time, was there any similarity seen or was there any congruence seen in the genetic material? So if you could see from this four population test, you can see that the Neanderthal and a present day European actually share their DNA at a given period of time. Unlike the present day African, where, as you can see from the A, it's different. So even a Neanderthal and a European can have similar DNA at a given period of time, similar DNA letters at a given period of time, but not an African. And what does that tell? So we did talk about the common ancestor thing. So if two genetic lines share a common ancestor closer in time, their genetic material is going to be similar. But if there is a difference in their genetic letters, That means that the common ancestor existed long, long time back. So that's what the four population test also revealed. That in case of modern, sorry, yeah. So all non-Africans today show some amount of Neanderthal DNA, while the Africans do not, which means that after the modern humans came up and they separated from the Neanderthal line, the Europeans, so the European Homo sapiens would have bred with the Neanderthals, and that's where the mutation occurs, not the African Homo sapiens. Um, Am I clear? Okay, this is not how we are supposed to do the lectures, but since it's just for research wing, uh, this is where the Neanderthal admixture was discussed about. So what we have now with the four population test is that yes, the Neanderthals did interbreed with modern humans. These modern humans were not the ones whose ancestors are present day Africans. Sorry, whose successors are present day Africans. These were Europeans, and so the European DNA shows the largest number of Neanderthal ancestry within them. But what was not revealed in this study is when exactly did this mutation occur, or when exactly did this interbreeding occur. So what the researchers did to analyze this problem was they utilized a technique called recombination. What is recombination? So as you can see, how do genetics work? The very basic function. You get genes from your great grandparents, uh, and then your grandparents, parents, child, and you will be transferring them to your own offsprings. So that's how genetics works. So think about a very long temporal span. Think about a time, let's say 39,000, 40,000, 50,000 years ago. DNA sequences have been going on since that time. So if I could take a modern human's DNA and reconstruct DNA going back to 50,000 years ago, I can see when the Neanderthal DNA got, like when it did show its presence in the DNA sequence. So, for example, this is the above-most, the dark gray bar is a Neanderthal genome. So it's purely Neanderthal. It does not have any element of admixture present in it. But if you see let's say the Neanderthal would have interbred with a Homo sapien from Romania. So you can see that after 100 to 200 years, the Neanderthal DNA would still be present, but it will be fragmented. So the dark gray bars that you can see it's not one coherent bar now. It is present in just few fragments. And then almost 8,000 years after that mixture, if you see the DNA material of a Siberian, so you would see that the Neanderthal fragment has also reduced because the Neanderthals have now gone extinct, but the Homo sapiens are still reproducing amongst themselves, and the admixture that occurred 8,000 years ago, it is still getting carried on, but in just fragments and bits, because there is no new admixture taking place. Then you can see the DNA data of a present day Chinese person. So 50,000 years since the Neanderthal admixture took place, the Neanderthal DNA is present in just traces. It's not even fragments now, it's just traces. And so, what recombination does is it does take up these very fragmented pieces of a present day human being's concentration of DNA, and then it like turns the wheel of time back 50,000 years ago to see when from this very traced picture the full DNA can be seen. And so 50,000 years of genetic interbreeding can be seen. And when researchers did that, they found out that the admixture occurred 54,000 to 49,000 years ago. That's roughly the time since that's roughly the time when Neanderthals were beginning to disappear from the face of Earth. So you can now picture all of this in a more dramatic sequence. So 400,000 to 40,000 years. We know 40,000 years was the last Neanderthal seen, and maybe 10,000 or 14,000 years before the disappearing of Neanderthals, Neanderthals did breed with modern day human beings, and their offsprings. Um, their direct offsprings may or may not have survived. That is also something that we can discuss if you have a question afterwards. But I would not further complicate this here. I would just say that maybe 10 to 15,000 before the years before the disappearing of Neanderthals, Neanderthals did actually interbreed with the modern humans. So that's how the Neanderthal DNA gets into the DNA of us or of modern humans. And why did this occur is something prone to a number of interpretation objectives. It could be a peaceful interbreeding or maybe a community cohesion that may have existed. That is something that can be constructed to, through archaeological study as well. It may have been a more conflict-like situation as well, which is that entire Harari's argument of colonization and of killing of Neanderthals and everything. But I'm not going into the why part. I'm just going into the what and a how part of how DNA was able to tell us that yes, admixture did occur, and when did admixture occur. So this was about the Neanderthals. Let us talk about another species of human beings that was discovered quite recently and has created a spark in the Paleolithic world or even in the prehistoric world. So Denisovans is again an extant species of the genus Homo, and they lived about 285 to 25,000 years ago. And since the skeleton. It's not even a skeleton. If you actually search about Denisovans, you would find out that it was just a finger bone that was discovered, and that's the only thing you got. And as we also discussed in the last lecture with ma'am, um, that's how genetic studies were conducted on just a fingertip, and that was the only DNA found. So it was found in the Denisovan cave of Altai Mountains in Siberia in 2008, and the Denisovans frequented your Eurasian the continent, um, continents of Europe and Asia. So when this DNA sample was compared to the DNA sample of Neanderthals and AMH or anatomically modern humans, that's us or the first species of the genus Homo, the mitochondrial DNA studies showed that the population of Denisovans had branched far before the human and Neanderthal divide actually occurred. So you have humans and then you have Neanderthals, but the ancestor of humans and Neanderthals would have branched off from the Denisovans long ago. Whole genome DNA analysis, not the mitochondrial, but the entire DNA studies revealed that Denisovans were more closely related to Neanderthals than they were to the modern humans. So, what was interesting in this study was uh, Denisovans, although the Denisovan content is far less present than the Neanderthal content in us, also because Denisovans and modern humans have a more distant ancestor as compared to Neanderthals. Still, researchers took DNA samples from modern populations and they compared it with the Denisovan DNA, and they found that interestingly, the closest of DNA that could be found was with the New Guinean population. So New Guinea is here. It's east of Australia. This is where the island is. And as you can see from the pie charts, the largest of DNA content of the Denisovans was found in the New Guinean population, present in New Guinean population, which is 3 to 6%. And that is the largest of the Denisovan DNA that survives in any present day human population. And again, when this recombination thing took place, when they did it on the New Guineans and Denisovans, they found out that their common ancestor would have existed 59 or 44,000 years ago. So it's likely that 60,000 or 50,000 years ago, the Denisovans would have had interbred with the ancestors of the present day New Guineans, and that's how the Denisovan ancestry emerged. in present-day New Guineans. Um, it's very important to understand that Siberia is in one part of the world, New Guinea is in another part of the world. So, obviously, the other question that baffled the researchers after this discovery was that where did this intermixing occur? So, did it happen in mainland Asia? But if it did happen in mainland Asia, so why don't we have so much of DNA of Denisovans present in us? Why do the New Guineans have much more DNA? So, other possibility was what if the intermixing took place in the islands? But the islands do not show evidence of Denisovan activity in any sort of archaeological record. There is nothing of Denisovan fossils or bones found in the islands. So, where exactly did this admixture took place? Now, here is what we go a step ahead or a level ahead. What researchers interpreted with this situation was that 60,000 years ago, Denisovans were definitely larger in number, definitely more mobile. They were frequenting different parts of Asia, and they were a diverse population, which means that there would have been further subspecies of Denisovans also present. One branch that may have existed in Siberia, and the other branch that may have existed in a much more southern area, and they would have bred with the ancestors of the present day New Guineans. And this other population, not the Siberian Denisovan that was found in the Altai Mountains, but the Australo-Denisovans would have been the ones who would have given contributed to the DNA of present-day New Guineans. These were called Australo-Denisovans, and it is a ghost population. So, what is a ghost population? A ghost population is a population that would have existed somewhere in the DNA chart or in DNA history, but their direct DNA sampling cannot be done in any case. That means Australodenisovans, they may have existed at a particular given time, but today we do not have any bones or fossils or anything to extract DNA from them to understand more about them. It's something that has been only constructed because of DNA sampling from other species or subspecies that a ghost population was identified. There are a number of ghost populations that exist and have been found out in genetic studies. Um, we can discuss this again towards the end, but as you would know by now, I would finish this segment here on Denisovans and would now go on to another very important DNA study. This is a landmark study. And this is not something that I say, this is something because it brought together genetics, isotopes, linguistics, as well as other sorts of archaeological evidence together. So, this is what we call quite literally all roads lead to Rome kind of study. The Proto-Indo-Europeans. So who were they? Um, when principal, okay, when uh, Eurasians, so people from Europe and Asia, the present-day Eurasians, when their DNA was taken up and it was put on a Cartesian plane. So this is what your Cartesian plane is on a graph. So what the researchers found that there was a European cline and then there was a Near Eastern cline. So cline is basically like a coming together of different genetic materials. So you can see that these lighter gray ones are the Europeans, and here the other lighter gray ones are the Near Easterners. These are the present day populations. And so because they have commonality in their DNA, they formed a row, an entire row, which is called a cline. Um, in order to understand migration in the past 5,000 years of European history, what the researchers did that they planted the DNA obtained from 800 present-day Europeans and Near Easterners, and they obtained these clines. To this, they plotted DNA obtained from ancient populations. So, the DNA of the first European hunter-gatherer, the DNA of the first European farmer, and of the later European farmers. And these were then planted on the same graph. So, this graph, you see that the black dots, not the gray dots. The gray dots are the present day population, but the black dots are the recent population. So when they were planted, it was shown that around 10,000 or 9,000 years ago, the, as you can see from here, the European farmers actually showed a lot of similarity or affluence towards the Near Easterners. And the researchers were finally able to understand that these first European farmers came from Anatolia, which is present day Turkey. So the first farmers of Europe were not actually the hunter gatherers of Europe, or as you would expect in a more evolutionary manner, that hunting gathering gave way to your farming. That's true. Hunting gathering gave way to farming, but the hunter gatherers, the European hunter gatherers, did not become the European farmers. The first farmers actually came from Anatolia or from Near East. And what when the researchers went further in this study, it showed that the first farmers actually retained their genetic material. That means they did not interbreed with the hunter gatherers they were encountering while they were living in Europe 9,000 years ago. But by 6,000 years ago, they actually started interbreeding. And so the farmers that you would see 6,000 years ago in Europe were actually a combination of the European hunter gatherers and the Anatolian farmers who came 8,000 years ago or 9,000 years ago. So, what we do get here that the European hunter gatherers were not Europe's first farmers. They came from Anatolia, and they did not immediately intermix with the local population or the existing hunting gathering population. They retained their genetic material. They were maybe a close knit, a more endogamous clan, and then gradually 2000 years ago, 2000 years later, they started mixing with the hunter gatherers. So this was what was revealed through this Klein or through this Cartesian plane plotting. This is called principal component analysis, which is also something important in DNA and isotope studies. It's nothing but a common element, a principal component, as the name says. So in this case, DNA, which was plotted and was sought to be understood. We can take this as a question as well afterwards. Now, let us come to the Proto-Indo-Europeans. Your Proto-Indo-Europeans are not these farmers who came from Anatolia or the hunting-gathering individuals. These are actually pastoralists who came 5,000 years ago from the highlands of Iran. So from Iranian steppes, these people came, and this was an entire migration that took place from Iran to Western Europe 5,000 years ago, which marked the beginning of the Yamnaya culture. So it's getting a bit complicated, but let's think about this: that if you meet a European, a present-day European, and you take out their DNA, you will find that they have DNA of Europe's hunter-gatherers. They have elements of those Anatolian or early farmers, and then they also have the DNA of Iranian steppe dwellers that came 5,000 years ago. So there was another migration that took place, and this migration 5,000 years ago that took place is crucial because they actually outswept the earlier farmers. They brought back pastoralism. So again, it's not a linear, history isn't never linear, but pastoralism became a more dominant form of subsistence 5,000 years ago in Eurasian history. And this is also shown in the archaeological culture as well as in isotope studies. So if you remember this paper that was discussed last time in the strontium presentation, so the migration, when it took place, how did the researchers come to this conclusion? So first of all, when they looked at burials 5,000 years ago, forget everything else. Let us go more chronologically. So as an archaeologist, I stumble upon burial that may have taken place 5,000 years ago. I find that the individuals are morphologically different from the ones that I saw maybe 6,000 years ago, 7,000 or 8,000, 10,000 years ago. So yes, morphology is different, but then morphology cannot really give us a more cannot tell a lot about an individual. But then I also discovered a distant sort of pottery, the bell beaker pottery present in the burials. I also see that the burials are not similar as they would have existed 6,000 or 7,000 years ago, because 5,000 years ago, the burials that I see are in form of these mounds. These are called kurgans. So, as an archaeologist, we are going chronologically. I've not conducted any isotope study, any DNA studies as yet. I've only seen morphological differences in the burials. I've only seen differences in archaeological material, that is in pottery, in the kind of burial that is present. I do not find any elaborate dwellings, any cities or any settlements that I found actually 6,000 years ago. So what does it tell me? It does tell me that a different population may have come 5,000 years ago. So there is a possibility of migration that may have taken place. But that's it. That's where I draw the line. How can I study migration? Now, we go back to what we just discussed 10 minutes earlier through isotopes. Through isotopes, when I take up 81 burials and I look at their strontium composition, and of the 81, I see that 51 of them have different strontium profile than the region they have been buried in. So they did not live in Central Europe. They came from some other place. So strontium suggests migration. You are one step ahead now. Now you are at this place that you know that yes, 5,000 years ago, there was a large scale migration that took place. Uh, the people came from somewhere else, and they were different. They had different pottery, different burials, they had different way of living. But where did these people actually come from? Again, this question still remains. This question now I will reconstruct with the help of DNA studies. So, when the DNA studies were conducted, taking in consideration of all the DNA material of present day Eurasians, Near Easterners, and Europeans, and in the past, it was understood that these individuals who came from somewhere else were actually coming from Iran and from the Pontic steppes. And they were the ones who brought this entire new culture. They were the ones who actually swept away the farmers or the farming culture that existed 6,000 years ago and gave rise to a more pastoralist economy and a more mobile settlement. So you don't have the large settlements, you don't have that sort of dominance on agriculture 5,000 years ago. Why? Because of this Yamnaya or the Proto-Indo-Europeans, they are the same. Yamnaya or the Proto-Indo-Europeans that came 5,000 years ago. I hope I was able to explain this well. I might have complicated this in trying to bring together the different elements of study, but as I said, we can discuss this again towards the end. So, linguistics, yes. I also said that it was not just the archaeological culture, strontium analysis, or DNA that told that yes, a migration took place. It was also understood with the help of linguistics. So, linguists like David Anthony, when they studied the Indo-European languages, they found out through the vocabulary. So, and they also used words like wheel or cart or horse. So, which talk about migration and movement, they found that these languages, the different languages that are spoken even in present day Europe, they all have their ancestry dating back at least to 5,000 years. So the commonality in these languages cannot go before that. And 5,000 years, we now know through strontium, through DNA, through archaeological literature, was when the migration took place. So I began this section by saying that all roads lead to Rome. And yes, this case study did show that did take place 5,000 years ago, a widespread migration, and it has been attested by linguistic studies, by DNA studies, by strontium or isotope studies, and by archaeological evidence as well. So, this is why this was one of the most crucial case studies that have taken place in the genetic history or the history of archaeogenetics. But there's always a but, and that's how I will be ending or concluding the presentation. Genetic studies can help us trace legacy, can help us trace origins, can help us trace migrations. But what are the larger interpretations we draw from them? What are the conclusions, and how do we present these studies? That's why one has to be very cautious when we are dealing with archaeogenetics, because if in the wrong hands, they can actually lead to a number of non-scientific or even scientific studies that can cause divides. And the best case study that I could think of was from India itself. So, three population test is similar to a four population test, except that there are three populations involved here. And so, in case of India, um, the Indian population, the present day Indian population of both North Indians and South Indians was compared with that of people from Europe and East Asia to understand to whom we were more closer or with whom we shared a more common ancestor. And we found out that Indians, all Indians, no matter if you're South or if you're living in the southern part of India or in the northern part of India, we all have West Eurasian ancestry present in us, except for the population living in Andamans, which have a more East Asian ancestry. But that's a different thing because the Andamanese population is a unique case in any sort of genetic study, for they have retained their genetic material for centuries, if not actually for millennia. So again, the principal component analysis. So the white dots here are the Indo-European speakers, and the black dots are the Dravidian speakers, which by default, if we talk about linguistics, can be categorized as the northern or the southern Indians. So the northern Indians, they show both of them actually, if you can see, have Western ancestors. If you look at the X and the Y axis, so all northern Indians and southern Indians have Western ancestry, except for the Andamanese, which are towards the right, and you can see that they have more Austroasiatic language as well as more Austroasiatic DNA in them. Now, I said this very casually because it was just the research wing today, but I would have been more cautious with the words I would have used had there been more people. And with that, you can already understand how difficult it is to talk about genetics for present day population, because genetics has been something that has been used for racial arguments for so long. Even when researchers, when they came with these DNA findings in case of India, they did not really wanted to say that the South Indians have less of the West Eurasian West Eurasian ancestry, while the North Indians have more of those uh, West Eurasian ancestry. So they came up with terms called ancestral North Indians and ancestral South Indians to not offend anyone. And they said that the ANI had DNA similar to Near Easterners and Europeans present day, while the ASI did not have any DNA that would have existed from population outside the country, which we know is not the case exactly, but it's a camouflage. It's a camouflage used to not create any linguistic racial divide. Um, when more analysis were conducted on the Asian DNA, or sorry, the Indian DNA, so the Y chromosome revealed that the Indian man and Near East, and the Eastern European man have a lot of similarities. So it can go up to 50% in certain cases, which means that yes, we did share common ancestors with them. But the mitochondrial DNA, if you remember, is something that comes from the mother's side of DNA, was largely restricted to India. So what does it say? That um, from the paternal side, from the side of men, there were Near Easterners coming in India, interbreeding with women of the country, producing offsprings, because of which we all have the West Eurasian ancestry in us. But if we talk about this in a more political sense or in a more social sense, it creates a power divide as well. That is something that I really do not want to use that term, but it has been so much used and so overused that we we would have guessed it. It's the Aryan invasion theory that is a very politically charged theory. So, did Aryans or did these Near Easterners, the powerful Near Easterners, interbreed with the women of Indian ancestors, or the local Indian women, and that is what led to us being the offsprings of them? Is a possibility that DNA studies actually suggest. Um, this is also seen in case of both the populations, so North Indians as well as in case of South Indians, because both of us have Iranian ancestry, and the first admixture occurred almost 4,000 years ago, which, if we put in the time frame, is around 2000 BCE. So we know that 2600 to almost 1400 BCE was a time when the Indus Valley civilization was in its mature phase, and by 1400 BCE, the civilization started crumbling. 2000 BCE, the first Iranian admixture took place with the Indian population at that time. I would not go further with that. I do not want to speak more on that because that just perpetuates that theory. Um, this DNA ancestry can even go further because all of us have the Near Easterner DNA present in us, and it can be reflected even in language and also in case of caste. So there are DNA studies that tell us about caste. It tells about the power balance in a given society. It talks about gender. It talks about languages. But it's very important for researchers to know how they are presenting these studies in front of people, and how are they using these studies, or what are they using these studies for. So we can again leave this here and just look at a few cases, because these are actual cases that have taken place, especially in American history, where Native American DNA in a number of cases was extracted, especially in the 20th century, without any permission. So it was stolen for research, and this has led to a number of questions being raised on racial integrity and integrity of genetics research themselves. So there are cases like these that have actually been they have been the cases have been filed on research organizations of, are they even taking permissions from the members of the community from whom they are extracting DNA, or are they even sharing these results with these people? And if they are not sharing these results, then for whom are these studies being conducted? And if they are still being conducted, then how are they even extracting DNA? Because we know that it's a destructive practice. So you can read on these cases. These are some of the more, sorry, these are some of the more known ones, which brings me towards the end of this entire brief presentation, that researchers have to be very, very critical of what they are trying to achieve through DNA studies, what they are trying to tell people, or to whom are they actually trying to tell these things. If you would want to read more on this, Gustav Cassina was a very famous archaeologist who supported the migratory model, and he was quite popular in 20th century, in early 20th century, and was one of the proponents of the migration versus the exchange models. And while he did conduct a number of important studies, his studies, all of them had a racial bias attached to them, which further helped the Nazis to develop their Aryan superiority theory. So we are well aware of how racism can be entangled with genetics or even evolutionary studies. So again, be very, very specific about what you're trying to talk about. Um, whom are you extracting samples from? Are you doing that in a more transparent way? Are you talking to the community members from whom you're extracting samples? And how are you publishing and sharing this research? In recent cases, especially in China, it was seen that I would urge you to maybe read on the Peking Man controversy, where the Chinese archaeologists, or in a number of cases driven by politics or national zeal, are trying to say that when the Homo erectus actually went out from Africa, um, the more believed model here is that the Homo sapien population first evolved in Africa, and that is how they spread to all the other parts of the world. While the other model says that no, it was Homo erectus who actually migrated to different parts of the world, and then regional migrations took place, which is not is believed by some archaeologists, but is now less agreed upon theory. Chinese archaeologists or Chinese archaeogenetics are trying to rewrite that history by saying that no, Peking Man or the Chinese Homo erectus actually gave way to the Chinese Homo sapien, which is distant from all the other Homo sapiens. This is a very biased theory. This is a theory that is rooted entirely in racism. But these studies are still being conducted, and these kind of information are being formulated through genetics. So, genetics again is it's a double-edged sword. It can help us understand about migration, movement, or even about our own population. But at the same time, if not used well, or if not used by for purely scientific research to aid the society, it can lead to a number of political problems. And with the wake of rising migration and wake of anti-immigrant emotions, it's very important to be crucial of our own studies. So that's it. That's the end of this brief presentation. And now I'm open to any questions or discussions that you guys would want to put forth.